Elizabeth Kinga Anopheles is an emerging multidrug resistant pathogen that has caused several global outbreaks. Within the E. anopheles NUHP1 genome, 14 antibiotic resistant genes have been identified, including ones required for resistant to aminoglycosides, beta-lactam, macrolides, tetracyclines, trimethoprim, and chloramphenicol. There are three types of chloramphenicol acetyltransferase enzymes or CAT, including A, B, and C, that catalyze the acetylation of the antibiotic chloramphenicol, thereby reducing its effectiveness as a viable drug for therapy. In the present study, we characterized the structure of CAT protein from E. anopheles NUHP1 strain that caused the Singaporean outbreak. We also performed a virtual screening of over 500 natural products driven from plants to find a potential CAT inhibitor. We used X-ray crystallography for structural determination. Additionally, we analyzed our protein in comparison with other CAT enzymes in terms of structure and sequence to determine the classification of our enzyme and putative active site residues. Finally, we use molecular docking method to find a potential inhibitor for the CAT enzyme. Our enzyme structure doesn't resemble that of classical type A CATs, but rather exhibits significant similarity to other previously characterized type B CAT enzymes from Pseudomonas aeruginosa and Vibrio cholerae. Based on the models that we made, the residues that could potentially hydrogen bond with chloramphenicol in the binding site of E. anopheles enzyme structure include proline 8, glycine 11, tyrosine 30, and serine 32 from one monomer and histidine 79 from a second monomer. These residues are all identical in the CAT enzyme from Pseudomonas aeruginosa. Putative residues critical for mediating hydrogen bonding interaction with acetyl-CoA in our structure are serine 139, lysine 162, and threonine 144. The residues are same in Vibrio cholerae cat B protein with different residue numbers. We also found asmanan, a polymannose present in the inner leaf gel of aloe vera plant, as a potential inhibitor for the CAT enzyme, acting as an antagonist in both chloramphenicol and acetyl-CoA binding site. Potentially, it can make 9 hydrogen bonds with the CAT enzyme in both binding sites. 6 of them are hydrogen bound with the putative active site residues. This would likely prevent both substrates to bind, thereby inhibiting CAT function. Our analysis of the E. anopheles CAT protein has shown it adopts a similar structure and retains conserved putative active site residues as clinically validated type B CAT proteins from other bacterial pathogens. However, further in vitro and in vivo experiments are necessary to show its activity and the inhibitor efficiency. Thanks for your attention.